specific slides that I can get back to that slide and try to answer.
Sir, I have a question regarding the mast cells and eosinophils. So you told that we know that eosinophils and mast cells both are, have the function of responding to allergic reactions. So could you explain what is the difference like in their role? Like,
Yeah. See, eosinophils are predominantly involved in allergic reaction, but of a particular type. Okay. So these are basically parasitic induced allergic responses. For example, if someone is infected with these worms like cystosomes, trypanosomes. Okay, so you actually find quite a lot of eosinophils in the circulation. Okay, mast cell as such are not so basically, how do they contribute to allergic reactions? Okay, so again, when we are going to talk about type 1, type 2, type 3, type 4 hypersensitivities, we are going to discuss in little detail. But for the time being, just remember that FC, these mast cells, okay, so they have on their surface these receptors. And what do they bind to? So these receptors are for antibody molecules, particularly the IgE type of antibody. Okay, again, I know that we really have not talked about different isotypes of antibodies, right? So there are 5 of those, and IgE is one class of antibodies which are predominantly involved in allergic reactions. Okay. Now, mast cells have these Fc epsilon receptors. Epsilon is basically the portion of the antibody which interact with the Fc receptor epsilon, right? Now, as a result of this, so you have these mast cells, you have this receptor on surface of these cells. This is a specific type of a receptor. It predominantly binds to this IgE, which has an Fc portion as a constant chain, which we call as epsilon. Okay. So now, so what you did was, so there are 3 stages of induction of allergic responses, right? In first, so this is called sensitization, and then induction, and then basically a long-term propagation, right? So what happens is these antibodies are formed. They are formed by your B cells, right? They are circulating, and wherever these IgE type of cells, someone has to, yeah. Okay, so now actually what you gotten in this allergic response is, so you have mast cells, and these mast cells are laden with these antibodies, which are now at this stage empty. Okay, empty means, so allergen is not there. Okay, so you have Fc portion of the antibody, Fc receptor. So this complex is already present on surface of these mast cells. Okay, now whenever you encounter this allergen, okay, dust mites, dust, pollen grains, other kind of allergies like drug sensitivities. Some people have drug sensitivities. For example, if some are given these penicillin, so they tend to develop allergic responses. So depending on what is that inciting cause, okay, so it will bind to the portion of the antibody. You know, antibody have this structure. This is basically the FAB portion, and then the stalk, which is the Fc portion, right? This Fc portion is bind to this receptor. This receptor is present on surface of these mast cells, right? Now, as soon as you encounter this antigen, it will bind here. So this will actually cross-link and it will induce signaling through this Fc receptor present in mast cells, and this causes degranulation, right? And whenever you have this degranulation, the stuff present in these cells, okay, like a lot of these granules are present, so which are carrying this histamine and other mediators, right? So those are released, and when these things are released in surrounding, it will attract more number of cells. So then you have all these symptoms of allergic reactions. Okay. In this case, mast cells are very critically involved. Okay. Now, what is the role of eosinophil? Eosinophils are predominantly involved in parasitic-induced allergic responses. All right. Rather, for any parasitic infestation, you normally don't call it as infection. You call it infestation. You find a lot of eosinophils induced in your system. But for normal allergic reactions, pollen grain allergies, dust, mite, household stuff, right? Drug-induced allergies. So for many of these things, so you have predominantly cell types which are involved in this process are mast cells. And because they carry in them these preformed granules carrying histamines and other active agents like prostaglandins and so on and so forth. And when these things are released, they are going to act onto your blood vessels. They are going to cause this constriction of blood vessels and so on and so forth. And then all these things will come into picture.
Fine, sir. And so another small doubt is that we know that monocytes and lymphocytes both are agranulocytes. So like all the myeloid-derived like these stem cells are, we know, granulocytes, except monocytes. So can we speculate any ancestral relation between these monocytes and lymphocytes based on the morphology, like because both of them are agranulocytes? So
well, yeah, you can say that. Well, ancestral relation. Well, if as I said, lymphocytes are part of your adaptive immune system, which is more advanced than your innate immune responses, right? Maybe, probably somewhere close to fishes. Okay. When the divergence of these fishes took place from the developmental history. Okay, so around that time, so you also started forming lymphocytes and the more defined subsets of the mediators which are involved in adaptive immune responses. Before that, so predominantly the responses are relying on myeloid cells, okay, which we call them predominantly be driving the innate immune responses, right? And now there are a lot of organisms which only and only rely on innate immune responses. They don't have actually lymphocytes and their products able to provide protection. Rather, they don't have the lineage development in lymphocytes. Now, your question is, can myeloid cells and at some time point might have originated from, for example, well, whether there is a relationship between lymphocytes and myeloid cells. Well, as such, these all these things are part of your defense mechanism. Obviously, we have to understand that. But in evolutionary history, in order to provide more diversification, you have to actually then fine-tune your responses to provide you the ability to defend more and more number of antigens, right? That was not probably required in the early form of lives. Well, I don't know whether actually that's the right way to put it, but what we know is more diversification, more antigen repertoire, more number of TCRs and BCRs. So they came later on and in the evolutionary history, and before that. So the defense of a host were largely relied on these innate immune responses, which basically have myeloid cells as their predominant effector cell population, right? You are saying whether lymphocyte and myeloid cells can also have the same lineage. Well, at some time, but they must have diverged from common progenitors. Okay, even if you go back, so all these cells actually are originating from hematopoietic stem cells. Well, obviously they have a linkage, right? But as you keep on requiring more and more fine-tuned defense mechanisms, so you keep on diversifying these different lineages, right? But as such, now we are kind of discovering that although if you think about it, for example, NK cells, which are part of lymphoid lineage, okay, but they are considered a part of your innate immune system, right? So that means there are always some shared features between these cells, and depending on their way of elicitation, how these cells get activated, what kind of protection they can provide, whether they can recognize limited sets of antigens, or they recognize these patterns, or so basically this is how you tend to divide your innate and adaptive immune responses, right? We already know that these four characteristic features are predominantly present in the cells of lymphocytes. Okay, that means they have a lot of antigen receptors expressed on the surface. There are ways how you can actually form these different types of antigen receptors, or which can be TCR or BCRs. But this is only the first line, okay, in the recognition part. If you go back little one to some extent, so the effector molecules are also more or less shared. Okay, so you have perforin granzymes and these molecules, surglycan. So these are all present in your myeloid cells as well as in your lymphoid stems, lymphoid cells, to carry out the effector function. But how these cells are triggered is different. And probably that is enough to provide you the diversification as these cells originate from bone marrow or other specialized organs to the well-organized structure where these T cells and B cells are formed. Always, you can consider that it's not like you have complete set of events to provide defense against one thing or the other thing. Okay, just remember the receptor diversity is provided by your lymphocytes, but many of the effector molecules produced by your lymphocyte and myeloid cells remain constant. Let's put it that way. That you always have some shared features, but you keep on giving more and more diversity, predominantly in form of these receptor recognition. And this is how you distinguish your lymphocyte and myeloid, or for example, macrophages, dendritic cells, other cells of your innate immune responses.
Fine, sir. Thank you.
Well, too much information, but so why we call as adaptive and innate is because the kind of repertoire they are able to provide you. Okay, so what kind of antigens you can recognize? All right, more questions.
Excuse me, sir. In the slide of NK cells, what was the third, like the third bit? What were the point that you were trying to make?
Which one? Here?
NK, the next slide.
This one?
No. Like 2 slides up. This experiment, this NK cells and tumors.
Okay, this one. So
One
here,
more, no, the 75th slide, sorry.
75th slide.
Yeah, the third point, the unmanipulated mice.
So you are asking, so what point I wanted to make using this slide?
Yeah, the
right?
third point, like what does it mean?
Okay, so the point basically says that in unmanipulated mice, unmanipulated means the animals which were not injected with a particular antigen, or they are not infected, or they are not developing any cancer. Okay. In these animals, if you basically take out this organ, spleen, and spleen, if you make, for example, a single cell suspension from spleen, splenic tissues, okay, those single cell suspension that you have prepared from these animals will exhibit some cytolytic activity. Okay. Normally, so this cytolytic activity is part of your adaptive immune responses. Okay, but that does not occur just like that. So first you have to induce, then activate your lymphocytes, and then basically these cells become effector cells, and effector cells will carry out this anti-tumor activity. Okay, now this is, all these things are not done. Okay, so you have not really activated your immune cells in these animals, but still you are able to get some tumor activity. Okay, and this was independent of this mechanism that you normally require for generation and activation and development of your T cells and B cells. Okay, and one of the enzymes which is critical for development of your T cells and B cells is RAG, recombination activation gene product. Okay, this is called RAG recombinase, right? Even if you eliminate this gene product from animals, so that activity was still present. One would like to think that if there is a killing activity, so you would like to attribute to the cells of adaptive immune origin. Okay, and if you already have knocked out this gene function, this RAG enzyme, so that activity was still there. So that means this was not contributed by T cells and B cells. Okay, And if you characterize these cells which carried this anti-tumor activity, so they were not expressing on them any of the marker which is associated with T cells and B cells. And that is why you call them as null cells, right? Now, these two things will eliminate that this activity which you recover from a naive, normal spleen collected from a mouse, which has not been injected with anything, which has not been infected, which is not developing a tumor. Okay, so how do you or which cells you attribute this anti-tumor activity? Okay, and how do you explain that anti-tumor activity? So that means there is some novel set of cells which are responsible for this activity, and they identified these cells as natural killer cells. Okay. Now, subsequent analysis revealed that these cells express these markers, NK1.1, many more markers. And now, for example, if you deplete these cells, specific depletion of these NK cells, as was shown in the next slide, something like here. So then this tumor activity was lost. And because of the elimination of those NK cells, so these animals, they kept on developing tumor. So this whole thing was to show that NK cells are these cells which are recruited in the response very early on. That's one. They have this ability to provide you some kind of anti-tumor activity. Their development is independent of mechanisms which form your T cells and B cells. Okay, so all these things will tell you that NK cells is a separate cell type apart from your T cells and B cells and can have anti-tumor activity. Yes?
Okay, sir. Okay. And also one more. What is the difference between chemokine and cytokine?
Well, okay. Cytokines, well, more or less, basically these are these protein molecules produced by your activated cells of immune system. Okay, cytokines. Okay. Now, chemokines have this critical role in recruiting cells from one place to the other. For example, you got an infection in your hand. So how do these cells come at that infected site? This is driven by the production of these chemokines. Okay, initially by those cells which are actually present in that local area. They produce these chemokines. There are many types of these chemokines, CCL2, CXCL2, CXCL10, and they basically recognize their counter structure present on the cells type they are recruiting there. This occurs in a gradient fashion. So, for example, at one particular place, if you have more concentration at one particular place, so you have less. So these cells will tend to move from a low gradient to high gradient concentration, and this is achieved by the activity of these chemokines. Okay, cytokines are a lot of effector molecules produced by these cells. Many of these cytokines also serve as growth factor for division and proliferation of, or sometimes there are some cytokines which are also responsible for their maintenance. There are so many of different types of cytokines which are produced by these cells, and same is the case with chemokine. One distinguishing feature between cytokines and chemokine is chemo attraction is brought about by chemokines, growth factors, proliferation, activation. So all these things are carried out by cytokines.
Okay, thank you, sir.
Okay, so more questions.
So, sir, can we say that cytotoxic T cells cannot work in conjunction with NK cells?
So what do you mean by conjunction? Because
So basically, NK cells and cytotoxic T cells cannot work together in destroying a particular infected cell type because of the missing self hypothesis that
okay. Well, you are probably right, and this is how these cells were at first place formed to provide diversification in the activity. Yes, because in order for your cytotoxic T cells to function, so you have to have MSC class 1 expressed. And as a mechanism where you downregulate MSC class 1 on surface of target cells because of their transformation, because of the cancer development, because of the infection of these cells. Okay, now you don't have MSC class 1, so then you also have to provide a facility where these cells do not go uncontrolled, and that can be brought about by NK cells. Because their recognition and activation is more or less independent of MSC class 1. Rather, MSC class 1 actually gives a negative signal to these cells, and if MSC is not present, so now you can have NK cells killing these cancer cells or infected cells.
So, sir, in that case, what are the conditions that we require for cytotoxic T cells to act against cancerous cells?
Well, you have to have, see, this is actually, it's not like, let's say a cancerous cell or infected cell. So they will always and always and always downregulate MSC class 1. No. So this is maybe a transient process, because when you have target cell means the infected cells or the cells undergoing transformation, right? During that process, when the cell is having this infection, okay, many of the products of these microbes or viruses, so they tend to help immune evasion process. And one of the mechanism by which they can do so is by downregulation of MSC class 1 transiently. Okay. In that case, your T cells will not be able to function efficiently, but your NK cells are still able to perform their function and carry out their effective function, right? Now, once for example, it's not like this microbe will keep on producing these products and it will keep on doing its activity, right? Maybe after some time, when these cells start expressing these MSC class 1, they start processing these antigens coming from these microbes and are loaded on surface of MSC class 1. So then your T cells can take over. Okay. So these are actually, you can say, the diversification mechanisms for controlling infectious agents or cancer cells.
Okay, sir. And can you go over the papain experiment of basophils once again?
Just a second. Where was it? Yeah, okay. So you have any specific points, or I just cover this slide?
The whole slide, sir.
All right. So in this paper, actually, what they showed was that these basophils can also serve as antigen-presenting cells. Now, for a cell to serve as antigen-presenting cell, so you have to have one classical feature, and that is the expression of MSC class 1, right? No, not MSC class 1, MSC class 2, because MSC class 1 is present on all kind of cells. However, if you can first demonstrate that on any cell type, MSC class 2 is expressed and is expressed significantly, this cell type is likely to have an antigen processing and presentation activity. Okay, so this is what they demonstrated in this panel. So basically what they did was, so they actually have this antibody against MSC class 2. Okay, now you can sort these basophils from these animals, and if you can stain with MSC class 2, okay, if you can demonstrate that these cells now express MSC class 2, so this is one of the features. Okay, and this is what they did here. So for example, so if you have this inactive papain is nothing but the antigen that was used. Okay, papain is basically it's a protein which is derived from papaya. Okay, so and this is used as a model antigens. This is one of the model antigens is used as papaya. Okay, papaya derived papain. This also has this enzymatic activity, and you will hear about its function during the antibody structure determination. Okay, so it can actually cleave specific residues in the antibody molecule. Anyways, so these cells were expressing MSC class 2 on their surface, and when you basically fed these basophils with active papain, more and more number of these cells started upregulating MSC class 2. And one other thing that you can actually very clearly make out from this slide is that staining for this MSC class 2 is not throughout in the sense in the cytosol and elsewhere. So this actually is present. So you have a cell, right? So more or less this MSC class 2 actually is present on the surface. And why that is required? Because those antigenic derivatives from this antigen, in this case, for example, papain. So those will be loaded onto MSC class 1 and displayed on surface of these cells. And when you do so, so these cells, these basophils, by coming in contact with T cells, can activate your T cells. These T cells will start making whatever the product they are supposed to do, and this actually is classified as the antigen presentation activity associated with basophils. So is that clear, or so you wanted to know, or maybe if you got a chance to read this paper, so, and if you specifically want to have a question on this. What I wanted to show you here was that basophils were shown to express MSC class 2 on their surface, and the class 2 expression was upregulated when antigen was added. And these MSC class 2 expressing basophils were able to engage your T cells to activate them. And all these features are associated with antigen presenting cells. Yes?
Okay, so basophils have this MHC class 2 on their surface, to which the papain gets interacted with, and then they form an interaction with T cells.
Papain derived peptides. Okay.
Okay.
Just remember, for MSC class 1 or class 2, so you don't present the whole antigen. Okay.
Yeah.
So the peptides which are generated in these cells as a result of this antigen internalization, and those peptides are formed, those peptides are loaded onto MSC class 2, and then they are displayed on surface to activate your T cells.
Okay, sir.
Yeah. So any more questions? Okay, if not, so maybe I think we have only a few minutes. So let's actually talk about this slide. So the purpose of this slide is that you can distinguish different cell types of your immune system based on what do they express on their surface, and this can be achieved through flow cytometry, okay, or even microscopically. Okay, you just have to stain these cells with an antibody against these molecules. Okay, let's say if you start with hematopoietic stem cells. Okay, so Vishnu has a.
So there's a question on the chat. Can you just see?
Can we discuss the previous quiz? So what specific question you have in this quiz? So you want to know basically what were the right answers? Just a second. So is that what you want to know, Mridula? Okay, the answer for first question was two diseases, rinderpest and smallpox. Immunology, Latin, immunus, exempt. Surveillance is achieved by the activity of your NK cells, which are part of your innate defenses, as well as your T cells. Okay, particularly involved against tumorigenesis. Okay, so so for example, if you have a tumor growth in your system, so that the activity of your T cells and NK cells is critically involved in providing you the surveillance. Immunological memory is associated with adaptive immune responses for both B cells, T cells. Yes, I know that we did say something about NK cells, but for the time being, we will call NK cells as innate immune cells. Okay. Now, why do you think memory is required? Well, you require memory to induce quick and efficient immune responses following a subsequent infection. Which of the following molecules directly act to control infection by coronaviruses? Interferon. Okay. Which of the following could act as PRR? And the answer is lectin-like molecules. All these things are PAMs. Okay. Lipotecoic acid, LPS, and PAMs are PAMs. Lectin-like molecules can serve as PRRs. TLR9 can recognize CpG, double-stranded DNA. Okay. Antimicrobial peptides create hole in the microbes. And these are all false, actually. The variolation and vaccination are not exactly the same term. Okay. The whole thing started from variolation, where they stole some material from cowpox lesion and injected but you don't call them as same. Vaccination and violation are not same. The word vaccine was coined in reference to horses. Well, actually, vaca means cow, so this is not horse. And obviously, the fourth one is false because the father of vaccination is Edward Jenner, or was. Yeah, lectin-like molecules are some of these receptors present on the surface of these cells. Yes. All right. So I think it's already 10:53. I think then probably we would continue with your lecture from tomorrow's class. So if you have more questions, so you can ask.
Sir, I have one small question. So what is the difference between lineage positive and lineage negative cells?
Okay, so I think you're talking about that slide I just displayed, right?
I think you also mentioned this thing in the staining slides, I guess.
Yeah, so just a second. Yeah. Okay, so lineage, this is basically the slide I think you are referring to, where basically you start from these stem cells and they are identified based on expression of these lineage marker. And one of the marker is SCA1, another is KIT or c-KIT. Okay, so if you can actually show the presence of these two markers on hematopoietic stem cells, you would say that these are the progenitor cells. Okay, now if these cells further differentiate into one lineage or the other, for example, all these molecules actually are your lineage determinants. Okay, particularly, for example, if you talk about CD3, CD3 is present on all T cells, so you would call this molecule as a lineage marker. Same is the case with B220. Okay, so this is also a lineage marker, because this will define that these cells originated from stem cells, but now these stem cells have already differentiated into this type, and now they start expressing another marker. Okay, so based on that, you would identify these cells as belonging to one particular lineage versus the other. Similarly, macrophage and granulocytes, depending on what do they express in terms of MAC1 or GR1. Okay, macrophages express low level of GR1 and MAC1 is high, and reverse in case of granulocytes. Megakaryocytes, which give rise to your platelets, they express CD41 as one of the markers, right? And erythrocytes you can identify easily based on this molecule that we call as TER119. So all these molecules actually are these lineage molecules. I think let's stop it, because you have to. There are only 3 minutes remaining, 4 minutes remaining rather. So you would be. You have to prepare yourself for the next class.